Hello, welcome everyone. I'm delighted to be here this rainy morning for those of us in Los Angeles. So my job today is to introduce our speaker, Dr. Christopher Bartley. Dr. Bartley completed his bachelor's and medical degrees at Yale and went on to also get his Ph.D. at Yale in neurobiology. He then joined us in California up at UCSF, where he completed his residency and postdoctoral fellowship in psychiatry. One of the things he did there was to oversee the immunopsychiatry clinic. And I think that clinical experience has really been, really carefully, closely connected to his research. So I'm excited to hear more about that today. In 2022, Dr. Bartley became a tenure-track clinical investigator at the NIMH, where he is currently the chief of the Translational Immunopsychiatry Clinic. So Dr. Bartley already, early in his career has received a number of awards. And to highlight just a few of those, he received the Edwin Alston Award for Leadership in Psychiatry at UCSF. He also received a very prestigious UC President's Postdoctoral Fellowship. He was a John A. Watson Scholar and was also a Hannah Gray Fellow at the Howard Hughes Medical Institute. Most recently, he's part of the Distinguished Scholar Program at the National Institute of Health. So I first had the pleasure of interacting with Dr. Bartley when he generously agreed to be part of a panel on psychoneuroimmunological perspectives on COVID-19 for a virtual meeting we did a number of years ago. He's got some really interesting papers in that area for those of you who want to be doing some research on his work after the talk. So I'm delighted to introduce him, have him join us today and learn about the current work that he's been doing at NIMH. Welcome, Dr. Bartley.
Thank you for that wonderful introduction, Julie. Let me go ahead and share my screen. Oops. Okay, wonderful. So it's so nice to be here with you all today. And I'd like to talk to you about the evolving concept of autoimmune psychosis. From the clinic to the laboratory. And so, as Dr. Bauer said, I'm the chief of the Translational Immunopsychiatry Unit. And one of the very cool things about coming to the National Institute of Mental Health is that when you start your own lab, you get to name your own lab. And so I chose the Translational Immunopsychiatry Unit. I have one disclosure. So in the past 24 months, I've served as a physician consultant for the Neuroimmune Foundation. So many of you have probably heard the term psychoneuroimmunology and are wondering how immunopsychiatry differs. And so as Carmine Perianti said in this clinical sort of introduction to immunopsychiatry, immunopsychiatry really highlights where our behaviors and emotions are governed by immune mechanisms rather than the other way around. And so in this sense, immunopsychiatry is really centered on the causal role of abnormal immune responses in behavioral pathology, otherwise known as mental illness. So what are the learning objectives for today? So we'd like to describe the origins of this evolving concept called autoimmune psychosis. We'd like to list the criteria for autoimmune psychosis as distinct from autoimmune encephalitis. We'd like to think about what tests we might order to look for autoimmune encephalitis and how to interpret those diagnostic tests that may aid in a diagnosis. And then finally, we'd like to explain how emerging research methods are expanding our understanding of CNS autoimmunity. So before we dive into the talk, I just want to provide some background information, and this will be completely familiar to many of you, but what is psychosis? I think we all know it when we see it, but there's not really a unified definition. And so one definition that I use is really that psychosis is a state of disordered information processing. And then there's really evidence that comes from multiple replicated studies that have identified neurophysiologic deficits in patients with the diagnosis of schizophrenia, the archetypal psychotic spectrum disorder. And so some of these neurophysiologic defects include pre-pulse inhibition, impaired sensory gating, blunted mismatch negativity, and deficits in efferent copy corollary discharge. And importantly, many of these neurophysiologic deficits are detected in the pre-attentive phase of information processing. And so these differences that we're seeing aren't a result of attentional differences or some sort of internal difference in some sort of conscious processing of an external stimulus, but really underlie some fundamental difference in how the brain is processing information. Of course, as clinicians, we can't really see these neurophysiological deficits. They actually manifest at the level of the individual as perceptual and behavioral problems. And so many of you are familiar with these categories that we observe and that we document clinically, that include hallucinations, delusions, disordered speech, and disorganized behavior. So for a while, even going back to Kreplin, it was thought that perhaps schizophrenia was comprised of multiple different subtypes. And over the past century, we've tried many times to subtype schizophrenia. However, those different subtypes have not necessarily led to any utility in terms of determining a therapeutic plan or identifying therapeutic targets. And so, as many of you know, in the most recent Diagnostic and Statistical Manual, number 5, schizophrenia was just sort of reaggregated in as a single diagnosis. However, I think that many of us still believe that at the biological level, at the etiologic level, schizophrenia is indeed very heterogeneous. And I think that we actually know that there are multiple causes of schizophrenia and more broadly psychotic spectrum disorders. Genetic studies have now revealed that about 2.5% of people with a diagnosis of schizophrenia have a large copy number variant that likely explains their diagnosis, the most common being 22q11.2 deletion syndrome, which is observed in about 1% of people with a diagnosis of schizophrenia. We also know that about 2.5% of people with this diagnosis carry a disruptive ultra-rare variant mutation, typically a protein truncating variant that leads to a non-functional protein. And these disrupting ultra-rare variants tend to occur in synaptic genes and chromatin remodeling genes. We also know that common variants likely contribute some to the risk, and there may be other genetic factors such as allelic variation in complement C4 and then environmental and substance factors. And so an open question is whether or not, given this underlying biological heterogeneity, a subset of individuals with a psychotic spectrum disorder may really have an autoimmune cause of their illness. So where does this idea come from? So in about the same year that Kreplin conceived this sort of construct of dementia praecox or precocious dementia, now known as schizophrenia, the future Nobel laureate Paul Ehrlich made a seemingly unrelated discovery, and that discovery was the antibody. So Ehrlich was investigating how the immune system learns to recognize foreign toxins and pathogens, and he theorized that these cells could recognize these microscopic interlopers via side chains, and that in response, these side chains would multiply and would seek out these microbes and clear them from circulation. And when he was asked whether or not these side chains might ever target the self, he responded that it would be dysetiologic in the highest degree if under these circumstances, self-poisons of the parenchyma, otherwise known as autotoxins, were formed. So ironically, in 1928, Ehrlich's own scientific descendant, Ernst Witebsky, announced that he had experimentally induced brain-reactive autoantibodies in rabbits. And this was an experimental paradigm known as experimental allergic encephalomyelitis or experimental autoimmune encephalomyelitis. And so within another 3 years, there were clinical tests that were being developed that used these anti-brain autoantibodies in an attempt to diagnose paralytic syphilis. So about 10 years later, Dr. Hermann Lehmann-Fausches, a German neuropathologist, developed what he called the flocculation assay for the detection of cancer. And this was performed by shaking healthy tissue with the bodily fluid, and if clumping of the tissue formed, it was evidence of an autoreactive antibody. And he used this as a diagnostic test for brain cancer. And he soon applied this diagnostic method to a range of neurologic diseases, including multiple sclerosis. And he found that 25% of MS cases actually had a positive flocculation assay. Surprisingly, in 1937, he screened the cerebrospinal fluid from hundreds of individuals with schizophrenia, and he reported that 95% of them had evidence of an anti-brain antibody. However, it was soon discovered that Lehmann-Fausches was unblind to the patient's diagnoses during testing, and his results were not easily replicated by other laboratories. And so as this autoantibody hypothesis fell into disfavor, the scientific community really set about to find an alternative biological explanation for schizophrenia. So if we move forward to the 1950s, psychiatrists had started to observe that reserpine and chlorpromazine, both of which inhibit dopamine signaling, were able to control agitation and attenuate psychosis without producing excessive sedation like many of the hypnotics that preceded those medications. And so this led to the hypothesis that hyperactive dopamine receptor activity could lead to schizophrenia. However, in the following decades, it was noted that D2 blockers were not totally effective in all individuals with the diagnosis of a psychotic spectrum disorder. And so a number of clinicians and researchers set about looking for alternative hypotheses. And so one product of the free love and altered consciousness movement was born in San Francisco, where I trained. And this was the street drug phencyclidine. So in contrast to amphetamine and psychosis, it was noted that PCP induced both the negative and positive symptoms of schizophrenia. And PCP, this observation really led Javitt and Zukin in 1991 to come up with this NMDA signaling hypothesis, or the glutamate hypothesis of schizophrenia. If we move forward now in time to 2001, the rheumatologist Betty Diamond, who was working at Rockefeller, demonstrated that some lupus autoantibodies are actually able to cross-react with and activate the NMDA receptor. And these antibodies bound to an extracellular portion, and in animal models would lead to memory deficits and behavioral changes. So she hypothesized that some antibodies to NMDA function as agonists, as the ones that she found, but some may also function as receptor antagonists like PCP and mediate lupus psychosis. So not many years later, in 2001, the neuroimmunologist Joseph Daumel really made a seismic discovery that many of you are probably familiar with, and this was the discovery of NMDA receptor encephalitis. And so he had identified 12 women with ovarian teratomas who developed subacute psychosis caused by inhibitory NMDA receptor autoantibodies. So 5 of the 12 women had been admitted to psychiatric facilities, and 4 of them completely recovered after immunomodulatory treatment. And so in that paper, he had a really important line. He said that these results emphasize the idea that autoimmunity can affect behavior, but the frequency of this disorder is unknown. And so this is, a really important point, and it's part of why we're here today. There's a growing recognition that the immune system can affect behavior, and therefore the immune system may serve as an entry point for treatment for subsets of individuals with an immunologic cause of their neurobehavioral disorder. And so since the discovery of NMDA receptor encephalitis, and this is really the first well-described autoantibody present in multiple individuals that have been reported in the literature that targeted a neuronal surface protein. Of course, paraneoplastic central nervous system disorders had been described before, in which the immune response was to intracellular antigens. And so if you look here at the timeline, you can see that there's been an increase in the discovery and description of autoantibodies that target both intracellular, but also extracellular surface antigens. So how do these autoantibodies work? And it's a simple heuristic that's not always true, but we tend to think that the autoantibodies that bind to extracellular targets are pathogenic autoantibodies, and they work through a number of different mechanisms. So in the case of NMDA receptor encephalitis, as you can see here, a single antibody is thought to bind to 2 NR1 subunits on 2 different NMDA receptor complexes. This leads to what is the word I'm looking for? In any event, cross-linking, excuse me. So the antibody, this leads to cross-linking of the NMDA receptor complex, and it accelerates endocytosis, and you get functional downregulation of NMDA receptor signaling. In other cases, such as antibodies that bind to aquaporin-4, which is the autoantigen that's targeted in neuromyelitis optica spectrum disorder, you see that these antibodies are able to fix complement, and you get formation of the complement complex. And this pokes holes in the membranes of glia in the optic nerve as well as in the spinal cord primarily. In the case of GABA B receptor autoantibodies, these can lead to direct inhibition of these receptors. And in other cases, you get autoantibodies that disrupt the interaction between a protein and its protein binding partner, as is the case with LGI1 encephalitis, which normally binds to ADAM22. In other cases, you can have autoantibodies that drive antibody-mediated cellular toxicity. And then there is emerging evidence that in some cases, these antibodies may actually be internalized, where they can then gain access to their antigenic target. So how are these autoantibodies discovered? So NMDA receptor encephalitis was identified by rodent brain tissue immunostaining. And so clinically, in order to diagnose autoimmune encephalitis, oftentimes a lumbar puncture is performed. And so that cerebral spinal fluid can be characterized using paraclinical testing, looking at cell count and protein and for oligoclonal bands. But on the research side, you can just do immunostaining, where you take a sagittal rodent brain section, you incubate it with cerebral spinal fluid, you wash that tissue section, and if there are antineural antibodies, they will bind to that rodent brain tissue, and then you can counter-label the bound immunoglobulin with a fluorescent marker. In this case, this is a 4088 marker. And so in doing so, you can start to pick up different patterns of immunostaining. And so here what you're seeing is a gallery of images that were produced by cerebral spinal fluid immunostaining of rodent brain sections from patients with various autoimmune encephalitic disorders. So in the top left, we have NMDA receptor encephalitis. And so anywhere you see dark brown staining, that indicates that there's antibody deposition in that part of the brain. And so here we can see that in the center of that rodent brain section in the top left, there's really intense dark brown staining. That's a structure called the hippocampus, which we all know is responsible for memory. And anterograde memory deficits are a core feature of NMDA receptor encephalitis. And you can see that there are subtle differences in the immunostaining pattern depending on what the antigen is. And on the bottom right, you can see what it looks like when there's no antineural autoantibody in cerebral spinal fluid. So this method is used both on a research basis, but it's now been incorporated into clinical testing. And so when you order the Mayo panel, you can order it for blood or cerebral spinal fluid. This is one of the test procedures that they'll perform. In fact, for NMDA receptor encephalitis, the first test they do is what's called the cell-based assay, which I'll show you in a moment. And if that cell-based assay is positive, then they actually determine the titer of the antibody using rodent brain tissue staining. So this is part of the clinical testing paradigm. So there are a number of methods that are used to confirm the presence of a clinically relevant diagnostic autoantibody. There are radioimmune assays where you perform an immunoprecipitation of a radiolabeled complex, and then you look at the radiographic signal that comes down with the patient's antibody. There are ELISAs, which is a colorimetric assay, and you get a change in the color of solution that's proportionate to the amount of autoantibody. And then there's cell-based assays. These are most commonly used in the Mayo panel I mentioned before. And this is where the antigen of interest is transfected into a cell. It's overexpressed, and then those cells are fixed and permeabilized. And you stain those cells with the patient's cerebral spinal fluid or their blood. And then finally, there are line blot assays, which are very similar to Western blots, where you have immobilized proteins along this strip, and then you incubate this line blot with the patient's biospecimen, and you look for immunoreactivity to this immobilized protein. So those are some of the test procedures that are used to identify and diagnose autoimmune encephalitis. And if we go back to the definition of autoimmune encephalitis, it's really changed over time. So prior to 2016, the most commonly accepted sort of criteria were that a diagnosis of definite autoimmune encephalitis, which sort of overlaps with this emerging concept of autoimmune psychosis, could be made if a known neuronal surface autoantibody was present in CSF or serum, and there was a response to immunotherapy. However, it was thought that these diagnostic criteria were really too strict because there was a lack of universal access to antibody testing. Antibody testing results can take several weeks to obtain. The absence of autoantibodies does not exclude a diagnosis of autoimmune encephalitis or autoimmune psychosis. We know that we're finding new antibodies on a yearly basis. And then a positive autoantibody test is not always diagnostic. Sometimes you have false positives in the post-infectious period or for other reasons. A response to immunotherapy is not available at presentation, and some patients simply may not respond to immunotherapy. So in 2016, Grouse et al. Worked together. This was a set of neuroimmunologists to establish a set of diagnostic criteria that allowed for the diagnosis of autoimmune encephalitis and really sort of definite limbic encephalitis as a sort of subset of autoimmune encephalitis in the absence of a positive diagnostic autoantibody test. And so in order to make this diagnosis of seronegative autoimmune encephalitis, all four of these criteria needed to be satisfied. So the first was a subacute onset of working memory deficits, seizures, or psychiatric symptoms suggestive of involvement of the limbic system. The second was bilateral brain abnormalities on T2 FLAIR MRI restricted to the medial temporal lobes. The third was either a CSF pleocytosis of greater than 5 cells per cubic millimeter, or an abnormal EEG showing epileptic or slow wave activity involving the temporal lobes. Of course, you could have both. And then fourth, a reasonable exclusion of alternative diagnoses, and these would include infections, sepsis, drug-related encephalopathy, cancer, et cetera. And so these criteria for the diagnosis of seronegative autoimmune encephalitis have really increased our detection of autoimmune encephalitis and have allowed clinicians to intervene much earlier. We're also starting to learn that CSF can actually be normal in autoimmune encephalitis. And so it's really important that this criteria number 3 is really either or. And so this was a study by Blinder et al., where they did a meta-analysis of cerebrospinal fluid clinical testing results across a number of different autoimmune encephalitides. You can see here they looked at GAD65 autoimmune encephalitis, LGI1, and moving over NMDA receptor encephalitis, AMPA receptor, and GABA B receptor encephalitis. And they looked at the proportion of these individuals with definite antibody-positive autoimmune encephalitis and asked what proportion of them actually had a pleocytosis. And you can see that it varies by type of autoimmune encephalitis. And in some cases, as in the case of LGI1 encephalitis, more than half of the patients have no evidence of an increased white blood cell count in their cerebrospinal fluid. Increased protein in the cerebrospinal fluid is another paraclinical marker that we use to determine whether or not there's a possibility that there's CNS inflammation. It's a pretty non-specific marker. Nonetheless, it's part of your standard testing when you perform a lumbar puncture and send the CSF for profiling. And here they looked at the proportion of individuals that had increased protein. And once again, you can see that it varies by diagnostic category. But in many of the cases, these patients have a normal protein. One of the CSF tests that should be performed every time, but oftentimes is not performed, is what we call oligoclonal banding. And some of you may have heard of this. And so oligoclonal banding is a technique whereby a sample of cerebrospinal fluid and a sample of serum are separated by isoelectric focusing point, and then is immunostained for IgG class immunoglobulins. And here what they're looking for are the presence of antibody populations that are selectively present in the cerebrospinal fluid, indicative of what we call intrathecal antibody synthesis. And so typically there's no antibody synthesis in the central nervous system. And so if you find antibody populations that are present in the cerebrospinal fluid, but not in the periphery, it really indicates that there's a B-cell mediated immune process going on in the CNS, and it's supportive of either an infection or some autoimmune process. And so there are 5 different types of oligoclonal banding patterns. Here on the left, you see type 1, where there are no antibody populations detected in either cerebral spinal fluid or the serum. Type 2, where you see CSF-restricted oligoclonal bands. Type 3, where you may see some CSF-restricted bands and some mirrored bands. Type 4, and this is what you often see in systemic autoimmune disorders like lupus and sarcoid, you can see a mirrored pattern of antibody populations. And then type 5, where you actually have a monoclonal antibody population that appears banded just because of minor sort of biochemical differences in their glycosylation patterns. And you might see this in something like multiple myeloma or MGUS. So Blinder et al. Looked at the proportion of individuals that had abnormal CSF findings based upon a combination of pleocytosis, total protein elevation, and the presence of oligoclonal bands. And once again, you can see that it varies by type of autoimmune encephalitis. For example, in LGI1 encephalitis, more than 50% of the patients have normal cerebral spinal fluid, whereas in NMDA receptor encephalitis, invariably, there's at least one CSF parameter that's abnormal. And so this concept of seronegative autoimmune encephalitis and the idea that patients with definite autoimmune encephalitis, antibody-positive autoimmune encephalitis can have normal CSF contributed to this emerging concept of autoimmune psychosis. And the sort of stated goal of this concept that was put forward by Tom Pollock et al. Around 2020 was that perhaps we're missing cases of autoimmune encephalitis because they're presenting with primarily psychiatric symptoms. And so by softening the criteria, we might be able to pick up on these individuals a little bit earlier than we might otherwise be able to pick up on them. And so they came up with these categories of possible, probable, and definite autoimmune encephalitis or autoimmune psychosis. And you can see that for possible, all you need is an abrupt onset of symptoms, less than 3 months, and at least one of these predisposing factors: tumor, movement disorder, adverse response to medications, neuroleptic malignant syndrome, severe cognitive dysfunction, decreased consciousness, unexplained seizures, or autonomic dysfunction. For probable, the requirement is that you have an abrupt onset of psychotic symptoms and at least one of the following: an increased CSF white blood cell count, MRI changes, or oligoclonal bands, increased IgG index, which is a measure of the ratio of IgG in the CSF relative to the blood corrected for albumin, which sometimes can be an indicator of intrathecal antibody synthesis, even in the absence of CSF-restricted oligoclonal bands, EEG changes, or a positive serum neuronal autoantibody. And then finally, for definite autoimmune psychosis, the above criteria needed to be met, in addition to a positive finding of a CSF neural autoantibody. And so these categories were supplemented by what are called yellow flags and red flags. And so these were clinical sort of features that might increase your suspicion that perhaps someone should be evaluated for possible, probable, or even definite autoimmune psychosis. And so on the left, you can see that the yellow flags are relatively nonspecific, and you might see some of these in your patients with sort of primary psychotic spectrum disorder. Nonetheless, these are considered to slightly increase the probability that someone may have autoimmune psychosis. At the bottom here, you see that there are common autoantibodies, anti-nuclear antibodies, and anti-thyroperoxidase and anti-thyroglobulin autoantibodies that are included in the yellow flag category. And then in the red flag category, you see infectious prodrome. So importantly, NMDA receptor encephalitis was first described in women with an ovarian teratoma. However, we now know that a large proportion of individuals don't have an underlying malignancy, and many of them actually have an infectious prodrome. We know that one of the most common causes of NMDA receptor encephalitis now is a prior diagnosis of HSV encephalitis. And then you can see that there are other red flags. I'll just point out short-term memory impairment as one of the most important red flags to pay attention to. So as clinical testing has continued, we've revised our thinking around some autoantibodies that were previously used as diagnostic autoantibodies. Importantly, the voltage-gated potassium channel autoantibodies are now no longer considered diagnostic. And so we know that what were thought to be VGKC antibodies that were associated with profound neurologic symptoms that were oftentimes immunoresponsive were really due to autoantibodies that target VGKC complex proteins LGI1 or Caspr2. And that there's a population of individuals that actually have an antibody that's bound to a molecule that's used to detect the VGKC protein complex rather than antibodies to LGI1 or Caspr2 or a potassium channel itself. So despite the increase in the number of classified autoantibodies over time, I showed you that chronology of autoantibodies are now considered to be diagnostic. Still, there's a large proportion, maybe 30 to 40% of individuals who present with what is classified as seronegative autoimmune encephalitis. And so clinically, they meet criteria for autoimmune encephalitis, yet there's no diagnostic marker. And so to try to support decision-making, the field has started to look for other indicators of autoimmune psychosis that might inform treatment. So neurofilament light has been reported in a couple studies. And what has been shown is that neurofilament light levels above 15 picograms per milliliter are predictive of NMDA receptor encephalitis instead of schizophrenia in the acute phase. Also, there's a PET scan, PET CT, is being used quite commonly in Europe. And what we find is that abnormal asymmetric or hypermetabolic or hypometabolic FDG PET is abnormal in about 82% of individuals with autoimmune encephalitis. And there is some evidence that some individuals who may meet criteria for possible autoimmune psychosis, but not autoimmune encephalitis, may also have abnormal PET scans and be responsive to immunotherapy. So an open question is, does autoimmune psychosis explain chronic schizophreniform disorders? Of course, when NMDA receptor encephalitis was first described, it was noted that the age distribution profile for NMDA receptor encephalitis looked eerily similar to the age profile for the onset of schizophrenia. And so as a result of this, there were a number of studies that were performed to look for whether or not we were missing people who may have an attenuated form of NMDA receptor encephalitis who were misdiagnosed with schizophrenia. Most of those studies have turned out to be negative, as I'll mention in a moment. And so this is a really important question because we know that about 1 in 150 people worldwide are diagnosed with schizophrenia. There's a huge economic burden. There are very few total pharmaceutical innovation dollars that go towards schizophrenia and the broader sort of psychotic spectrum disorder category. And although this may change soon, there have been zero first-in-class medications since the introduction of Clozaril in 1989. And so before we talk about the presence of NMDA receptor encephalitis antibodies in people with schizophrenia, let's take a look at the overall CSF profile in individuals with psychotic spectrum disorders. And so these data are aggregated from a number of large studies that came out of Germany, where now it's part of the clinical recommendations that anybody who presents with first episode psychosis be offered a lumbar puncture. And so I think in aggregate, these studies have looked at maybe 1,500 to 2,000 individuals. A couple of them have been reported by Dominic Endres, and then another by Alkomi Hassan. And what they've shown is that nearly 4% or 40% of these individuals have elevated IgG in their cerebral spinal fluid. However, if you look for classified autoantibodies, only 0.3% of them have a classified autoantibody consistent with autoimmune encephalitis, indicating that we're not really missing many of these cases in the population of people with chronic psychotic spectrum disorders. 1% of all Ph.D. cases harbor an unclassified autoantibody in CSF. And if you actually index on red flag cases, who may be atypical in some sort of aspect of their clinical presentation, up to 20% of those cases harbor an unclassified autoantibody. So that data really comes from this large study from Dominic Endres, where he looked at nearly 1,000 individuals with a schizophreniform disorder or an affective psychosis. And he found that 2.5% of these individuals had autoantibodies, but only 1% of them had unclassified autoantibodies in cerebral spinal fluid. And the way that he detected these unclassified autoantibodies was by doing rodent brain tissue immunostaining, which I presented to you before. What was really fascinating about this study was that 2/3 of these individuals with these unclassified autoantibodies in cerebral spinal fluid who were offered immunotherapy were actually immunotherapy responsive. And so this has been followed up in subsequent studies where they focus on patients with schizophreniform disorders with red flags. And so in this study from Dominic Endres, they looked at 70 individuals, and 2 had known autoantibodies, one against SOX1, the other against an astrocytic protein, GFAP. And then in cerebral spinal fluid, they found that 20% had CSF immunoglobulin that was reactive to rodent brain tissue, and that 2% of these individuals had white matter autoantibodies. Here you could see the CSF profile for these individuals. So 5% of them had a pleocytosis, 33% had an elevated protein, and 9% of them had CSF-restricted oligoclonal bands. In the healthy population, this is typically between 0 and 4%. And once again, 9 of 12 of these individuals were responsive to immunotherapy. So during my postdoc, I wanted to ask a similar question. I worked with Michael Wilson and Sam Pleasure at the University of California, San Francisco. And Michael Wilson had put together a large cohort of individuals with undiagnosed neuroinflammatory disorders, some of whom had prominent neuropsychiatric symptoms. And so in that biobank of about 1,200 individuals at the time, there were about 400 or so who had some evidence of psychosis, at least one psychotic symptom domain. And so we took the cerebral spinal fluid and immunostained rodent brain tissue, and were able to recapitulate those findings that Dominic Endres had published. And we found that about 20% of those individuals had CSF antibodies that were immunoreactive to rodent brain neural tissue. And this was true across all ages. And so you can see the histogram of the age distribution on the bottom left. So some of these patients really did look like they had autoimmune encephalitis. And so this is the CSF immunostain that was produced by this CSF of a girl who was 16 years old, who presented with acute onset psychosis and catatonia, as well as autonomic instability. And her presentation was steroid responsive. And what you can appreciate here, this is a rodent brain. So on the left is the front of the brain, that's the olfactory bulb, and on the right is the cerebellum. You can see the brainstem down below, and this should you should not see any green. And so anywhere you see green is indicative of antibody binding to rodent brain tissue. And you can see that there's bright green in this lower panel along the border of the brain. These are the meninges, as well as some somatic staining within the parenchyma of the cortex here, as well as some staining of the neuropil. But we also found evidence of autoantibodies in the cerebral spinal fluid of individuals who did not meet criteria even for possible autoimmune psychosis. And so this is the CSF immunostain pattern that was produced by a 42-year-old woman. And actually, this is a different person, but an individual with schizoaffective disorder, depressed type. You can see that the immunostaining pattern is remarkably different, but there are antibodies that bind throughout the brain. And so this really led to this question, although it doesn't seem that we're missing many cases of autoimmune encephalitis in our population of individuals with chronic psychotic spectrum disorders, there's still a proportion of them that have abnormal CSF findings. And that proportion is higher than you would expect to find in the healthy population. And so our clinical tests really only test for about 20 or so neural antigens. And might there be a way on a research basis to actually screen for antibodies against the entire human proteome? And this is important. We know that the humans have about 20,000 genes that encode for hundreds of thousands of proteins, which can actually encode millions of different epitopes to which autoantibodies can bind. So might we be missing some cases of autoimmune psychosis simply because the clinical tests have blind spots? And so I've told you about immunostaining, but one of the techniques that I learned with Drs. Wilson and Pleasure during my training is called phage display immunoprecipitation sequencing. And so bioinformatically, we broke up all known human proteins and protein isoforms into peptides of 49 amino acids that overlap by 25 amino acids. And then we printed DNA oligonucleotides encoding those peptides and cloned those into T7 bacteriophage. And so a bacteriophage, if you think back to your basic biology, is a virus that infects bacteria. And in particular, the T7 bacteriophage can accommodate structural variation in its viral capsid. And so you can fuse a peptide onto the end of this 10B protein, and then the T7 bacteriophage will display 5 to 15 copies of a single peptide on its viral capsid surface. So in this way, we were able to clone the entire human proteome, about 750,000 peptides, into a single library of bacteriophage, and we could get 100x coverage in a volume of about 10 microliters. And so you can take that library of the human proteome on phage and incubate it with blood or cerebral spinal fluid. If there are antibodies that bind to those peptides, you can isolate the antibodies and the T7 phage to which they bind using AG magnetic beads, and you can wash away all the bacteriophage that aren't bound. And then you can subject the T7 phage that you elute off these beads to next generation sequencing, and then you can map those sequences back to the reference proteome, and you come up with a prioritized list of peptides and proteins to which the patient's antibodies might bind. So does this work? And the answer is that this technique does work. And so one of the sort of most ringing examples of this was in the discovery of a new autoantibody that is a biomarker for an unusual paraneoplastic form of autoimmune psychosis without evidence of an itis. So clinically, there isn't strong evidence of inflammation in most of these cases. And so I'll just walk you through an example case. This was a 5-year-old girl who was admitted for psychosis. And over the preceding months, she had gained 30 pounds of weight due to hyperphagia. Risperdal was ineffective. And on exam, she had precocious puberty and alveolar hypoventilation. So because she was so young and this was such an unusual presentation, there was an extensive workup that was performed. And you can see that MRI, genetic testing, infectious testing, standard paraneoplastic markers, and then the CSF autoantibody test, these were all negative. And so ultimately they fell back on a diagnosis of a primary psychiatric disorder. However, in the subsequent weeks, the child started to complain of GI problems, and this led to abdominal and thoracic CT, at which point they diagnosed an adrenal ganglioneuroblastoma. And she was diagnosed with this clinical entity at the time called rapid onset obesity with hypothalamic dysfunction, hypoventilation, and autonomic dysregulation. Unfortunately, this young girl passed away, and on an autopsy, even though there was no evidence of inflammation in her cerebral spinal fluid on lumbar puncture, they were able to find evidence of immune infiltrates in her hypothalamus, indicative of hypothalamic encephalitis. And so my colleagues, I was not part of this study, but my colleagues at UCSF were able to get biospecimens from 9 children who carried this clinical diagnosis of ROHAD. And what they found was that, in general, there was no evidence of pleocytosis, and there only in 2 patients did they find CSF-restricted oligoclonal bands. However, when they screened the cerebral spinal fluid and the serum of these patients on FIP-seq, they found that they uniformly enriched peptides that map to this protein called ZSCAN1. So ZSCAN1 is a transcription factor that's relatively recent from an evolutionary standpoint. It's not in rodents. And so if you stain rodent brain tissue with these patients' cerebral spinal fluid, it'll show up negative. ZSCAN1 does have a developmental expression profile, and its expression is highest in the hypothalamus. And so some of the lessons I think we can take away from this are that the clinical tests are limited and not dispositive. Behavioral pathology due to CNS autoimmunity can occur without a clear clinical itis, and that rodents don't express all relevant antigens. So in my own work, we found something similar. So we recently published an overview of autoantibody characterization in a syndrome called TRIM9, TRIM67 autoimmune neurologic syndrome. Most of these patients develop a cerebellar ataxia. A subset of these patients present with what looks more like a limbic encephalitis. Here I'm going to tell you about one of those cases that we applied FIPSeq to. So on the left is the immunostaining pattern that's typically produced by patients with TRIM9, TRIM67 cerebellar syndromes. And this patient was actually on a checkpoint inhibitor therapy for melanoma and developed cerebellar speech and ataxia, and unfortunately ultimately passed away. And then this patient on the right didn't have a known underlying cancer, did not have any cerebellar deficits that were detected on neurologic exam, yet was also positive for TRIM9, TRIM67 autoantibodies. And so we asked, why was this patient's clinical phenotype so different? This patient was a 60-year-old man who went to India, and about 20 days after arriving in India, started to sleep a lot. He began having anterograde memory problems. He started to overeat, and he gained a significant amount of weight over just a few weeks. He was ultimately taken to a local hospital where it was revealed that he had hyponatremia and his serum sodium was 121. And then on T2 flare, they identified that he had medial temporal thalamic and hypothalamic hyperintensities. Initially, he was treated with steroids and plasmapheresis, and he had a significant improvement and was brought back to the United States. However, 5 months later, he developed dream reality confusion, was more hypersomnolent, was sleeping 18 hours a day, and was noted to have blunted prosody. And he also had these really interesting egosyntonic auditory and visual hallucinations where he would be sitting in a room and would imagine his family coming into the room, dead ancestors, and they would be providing him encouragement and complimenting him. So here you can see a coronal T2 flare, and you can see that these arrows are pointing to the hyperintense regions around the third ventricle, the medial temporal lobes, and the upper brainstem here. And so we performed FIPSeq on our cerebral spinal fluid. And in addition to the TRIM9, TRIM67 antibodies, we found that there was a massive enrichment of peptides that mapped to this protein called receptor transporting protein number 5, and was enriched much more so than 5,000 controls and 1,200 patients with mysterious unclassified neuroinflammatory disorders. And so this enrichment was rare amongst the nearly 10,000 individuals that we looked at. We identified 3 that enriched peptides to RTP5, although we only have clinical data for one of these individuals, and this is the individual on the left. And so we validated these autoantibodies to RTP5 using a cell-based assay. So we transfected HEC293 cells with a construct that has a flag tagged RTP5 encoded in it, and that's shown in red. And then we fixed and stained the cells with the patient's cerebral spinal fluid in green, and you can see that those two immunostaining profiles overlap. So like ZScan1, there is no rodent ortholog for RTP5. It's relatively new from an evolutionary standpoint. And if you look at the Allen Brain Institute gene expression anatomic profile, you see that it does appear to be enriched in a pattern that's consistent with what we saw on the previous MRI. So you see the medial temporal lobes here, and you also see that the hypothalamus is bright red. So based on So although there's no rodent ortholog for RTP5, it is in a family of 4 other RTPs that are expressed in rodents. And so there's been some molecular biology that's been done to understand those. And it's now thought that as a class, these proteins are responsible for regulating G protein coupled receptor trafficking up to the plasma membrane. However, RTP5's anatomic expression pattern appears to be unique and does not overlap with the other RTPs, suggesting it may have a unique function. If you look in the literature, you'll see that this protein has not been well studied. There are no studies looking at the function of RTP5. And so we wanted to simply characterize its protein expression pattern, knowing that sometimes there are differences between gene expression and protein expression. So we did a 23 region anatomic immunostaining profile of the human brain. And so what we're going to focus in on is section C10 here. And once again, we're looking for brown immunosaying. Brown indicates RTP5 expression. And so C10 is around the third ventricle, and so this includes the hypothalamus. And so you can see that at this low magnification, there's sort of this haze of brown that abuts the ventricle on the left and is sort of throughout the hypothalamus. However, if you zoom in and you look at higher magnification, you see that this is sort of not just diffuse immunostain, that there's a subpopulation of cells. And these cells are most intensely stained in the tuberomammillary nucleus, which is a nucleus that's known to be rich with histaminergic neurons. And these histaminergic neurons are in part responsible for regulating wakefulness. And if we look at 100x magnification, we can start to make out the subcellular expression profile of RTP5. We can start to make out these little vesicle-like structures consistent with this hypothesis that perhaps RTP5 is responsible for regulating the trafficking of G protein coupled receptors up to the plasma membrane, but perhaps in a unique subpopulation of cells within the hypothalamus. So that's really interesting. These patients have, dramatic phenotypes. So perhaps it's not that surprising that we found an autoantibody in their cerebrospinal fluid, but are there autoantibodies in the CSF of patients with more prosaic forms of schizophrenia or schizoaffective disorder? And because I'm running short on time, I won't go through all of the cohorts that we're looking at, but I'll jump forward to one interesting patient that we're currently working up. And so this was a subject that was part of a biobank from Northwell Health. And it was part of a study that was overseen by Todd Lentz and Juan Gallego. And what we found was this patient had CSF autoantibodies that were tropic to the white matter. So on the left here, you can see negative immunostaining of a sagittal rodent brain section. This is the negative control. In the middle, you can, if you look carefully, you can make out that there's neon green staining of these white matter structures, so the optic tract, the anterior commissure, and the corpus callosum. And so this patient had anti-white matter autoantibodies. We performed immunoprecipitation mass spectrometry, and we identified 5 peptides that map to a protein called alpha-crystallin B that were exclusively pulled down by this patient's cerebrospinal fluid and not by the CSF of any of the other psychotic patients that did not have white matter staining, or control patients, or healthy controls, or control patients with neuroinflammatory disorders. And so when you look at the gene expression pattern on GenSat, you see that there's also it replicates what we saw with the immunosaying, where there's this increased expression in the corpus callosum, the anterior commissure, and the optic tract. And so we developed an assay, really a modified assay. Peter Burbello here at NIH has come up with a autoantibody validation tool called luciferase immunoprecipitation system. Uses vanilla luciferase. Ours uses nanoluciferase. And so this nanolips assay was used to validate the presence of these anticrystalline alpha B antibodies in this patient's cerebrospinal fluid. And so we always fuse this nanoluciferase molecule, which is an enzyme that produces light, to both the N terminus and the C terminus of our target candidate autoantigens. What was really interesting was that with the N terminal construct, there was no increased signal in the patient's produced by the patient's CSF, but in triplicate studies where we're able to demonstrate anti-Cry-A-B antibodies using the C terminal construct. So we decided to characterize this protein by Western blot, and you can see that for reasons that are mysterious, this N-terminal construct in green produces 1 proteoform, but the C-terminal construct produces 3 proteoforms. And we now know that the patient's autoantibody is selectively immunoreactive to the second of 3 bands in the C-terminal construct. This is interesting to us because anti-Cry-A-B antibodies are not necessarily new. They have been described in both multiple sclerosis and in systemic lupus erythematosus. However, uniformly in these disorders, these antibodies appear to target linear N-terminal epitopes and are not immunoreactive to the full protein. In contrast, our patient did not enrich these crystalline alpha B peptides by phage display, indicating that the antibodies don't bind to a linear unmodified epitope, but did pull out crystalline alpha B by immunoprecipitation, and which was validated by LIPS. So we think that the patient's antibody likely recognizes a post-translational modification, which indicates that this patient's anti-Cry-A-B antibody is likely categorically distinct from the autoantibodies that may be disease-modifying in multiple sclerosis. So I think I've covered a lot, but in summary, I want to say that autoimmune psychosis is really an emerging diagnostic construct, and red flags should lead to an extended workup. Clinical autoimmune psychosis is most commonly due to classified autoimmune encephalitis. Possible autoimmune patients with red flags may still respond to immunotherapy, as we saw in that large study from Dominic Endres. And the autoimmunity can really cause schizophreniform disorders, or at least psychotic symptoms, in the absence of significant itis. Finally, we think that emerging technology should improve our identification of autoimmune subgroups and may teach us something about neurophysiology, as hinted by the RTP5 data. And so here at NIMH, we're setting up sort of a dual approach to studying this phenomenon. So the Translational Immunopsychiatry Unit, which is my lab, is comprised of both a wet lab, where we recruit biospecimens from patients with all sorts of psychotic spectrum disorders, and we perform molecular screens for autoantibodies using a variety of methods that I've described today: FIP-seq, IPMS, protein arrays, and tissue-based immunofluorescence. And then we validate those autoantibodies, and we just recruited a protein engineer who's helping us to model some of these autoimmune disorders. But the goal is to do most of that to recruit the biospecimens and to do the discovery and validation in the wet lab. And then to do targeted recruitment of autoantibody positive individuals to the NIH Clinical Research Center, where we can do deep immunophenotyping, neuroimaging, and neurophysiology, behavioral phenotyping, and in some cases, actually treat these patients with immunotherapy and do convalescent phenotyping. So this is really happening on the pediatric side. And so we partner with Dr. Gina Mooneyham, who's the director of the Autoimmune Brain Disorders Program. And she's a triple-boarded pediatrician and pediatric and adult psychiatrist who is really focused on seeing pediatric patients with suspected seronegative autoimmune encephalitis. And then we're working to set up the same program on the adult side. We just saw our first patient here a couple of weeks ago, a really lovely 25-year-old man with a diagnosis of schizophrenia, but in the context of type 1 narcolepsy, a history of immune thrombocytopenic purpura, epidermolysis bullosa, then chronic recurrent osteomyelitis that was culture negative both times, suggesting that it may be autoimmune in nature. So he has this diagnosis of schizophrenia, but it really occurred in the context of multiple systemic autoimmune disorders, raising the possibility that perhaps his psychosis is autoimmune as well. So just wanted to quickly thank many of the people that helped me with my research during my postdoctoral fellowship at UCSF, and funding from the National Institute of Mental Health, NINDS, FNIH, HHMI, and the University of California system. And then just wanted to thank the current members of my lab here at the National Institute of Mental Health, and thank you for your time.
Dr. Bartley, thank you so much for a just very, very interesting talk. I've got a few questions coming in, and I'm also aware that we have like 2 minutes. So let me start with this one. Given limitations on sophisticated testing in the clinical setting, how soon in the course of treatment failure for new onset psychosis might you consider an empirical trial of steroids, with concern for the flip side of potentially steroid psychosis exacerbating the symptoms?
Yeah, so I mean, so typically, so I would say currently the recommendation is not for sort of empiric trial of steroids in the absence of some paraclinical laboratories that are suggestive of some underlying autoimmune process. So treatment failure alone is not sufficient to lead one to initiate steroids or immunotherapy. Now, catatonia or neuroleptic malignant syndrome that's severe, that's precipitated by antipsychotics, perhaps in the sort of context of some anterograde memory loss or some sort of focal neurologic deficit or orofacial dyskinesias, that would lead someone to a trial of immunotherapy. But we know that about 40% of our patients with a diagnosis of schizophrenia simply will not have a good response to antipsychotics. So that in and of itself is not sufficient to trial immunotherapy.
I've got another one saying, what a fantastic talk. Thank you. What are your thoughts on the overlap, if any, between psychotic disorders caused by autoantibodies and the excessive pruning hypothesis of schizophrenia that includes involvement from complement and microglia?
Yeah, it's a good question. So far, at least in the autoimmune encephalitic disorders, most of these antibodies that bind to neuronal surface proteins don't seem to cause massive complement deposition. I think the one example that I mentioned was neuromyelitis optica spectrum disorder, and that's really sort of an optic neuritis as well as a longitudinally extensive myelitis, and typically not associated with psychotic symptoms. And so, at least with autoimmune encephalitis, that's not really the sort of pathobiology that we see. Of course, these antibodies in some cases may fix complement. And so on the research basis, we'll oftentimes look at the subclass of IgG because we know that IgG 1, 2, and 3 can fix complement and IgG 4 cannot fix complement. I think that the genetics for complement C4 seem pretty strong. However, it's not clear how much increased complement levels in the cerebrospinal fluid of people with schizophrenia are actually contributing to their symptomatology. And so, I prefer, an alternative sort of model for most of these antibodies, which is that they are, directly pathogenic, that they're altering the targets, their molecular targets in some way.
Great. I think we're going to have to leave it there because we're going a minute past time. Thank you again for this fascinating work. We look forward to seeing how this progresses in years to come.
Yeah, thank you so much.